and welcome to the next chapter. And that chapter is about transcription. So I would want to use this class to take you back about where we started, and then have some introduction that will cover some points that will be required for you to understand the rest of the chapters. So we have learned about, to start with, we learned that DNA is the genetic material. That is what one of the first chapters we have learned. Then we have learned about the genetic organization, right? Then we have learned about how this genetic information can change. That is, we learned about mutations. Then we also have learned about recombination, which is also a part of change or mutations, but it is something like that happens by an intent. In the sense, I hope you understand the difference between these two. I will instead try to put this as mutation instead of writing as change, so that it will be more clear. Right? There is one difference between mutation and recombination. Both of these are actually change. In one of the cases, there is no control at all. Everything is absolutely random. Right? We have also learned about repairing and so on. Recombination, on the other hand, it's like it has evolved. It's a way of happening. Some enzymes mediate it. That means they are making, it is a willful, okay? It is anthropomorphism. Anthropomorphism is something like saying the bacteria didn't like this food, so it went to a different place. As if bacteria has a taste, as if bacteria has consciousness or self-awareness. That is called as anthropomorphism. In biology, when we are speaking, it becomes difficult. I think I have been emphasizing it all through the courses that we have been discussing, or the classes. So here, the difference between mutation and recombination is, in mutation, absolutely there is no control at all. It happens randomly because of external or spontaneously. Whereas recombination, we learned about two types of recombinations. One is homologous recombination, and the other one is site-specific recombination. Right? In homologous recombination, we need homology or sequence similarity. Otherwise, it would not happen. Right? Second thing, in site-specific recombinations, we need these recombinase recognition sites. Only there that's recombination occurs. So if you see, both these are mediated by enzymes. Right? In the change, what we're talking about mutation, it is kind of not within anybody's hand. It's not an evolved thing. It happens, and the cell has to produce enzymes to repair those mutations. Okay, so that is one of the difference you should keep in mind when you're talking about change in genetic nature. So that is what we have learned. DNA is the genetic material. We have learned about how the chromosomes are organized, types of genetic information, and so on. Then we have learned about change in the sense mutation and repair. We have also learned recombination, homologous recombination, site-specific recombination. We have also learned transposition. Now, what you should think of it as is, there is a chromosome, and we have learned everything as it is. Now, what can this chromosome do is what we are going to learn. It cannot, again, it's kind of anthropomorphic sentence. It's not that it has an intent or anything, but it is just things happen. There are several genes, and each of these genes should be expressed, and they will form mRNA, and which may give rise to proteins. And these proteins could be two types. There can be structural and they can be enzymes. They could be enzymes. If they are enzymes, they modify a product from reactants from A to B, and that the product may have an influence. It may release some energy and so on. That is how, if you are looking at genetic information perspective, everything in biology comes together. Okay, everything is kind of destiny, right? And that destiny is the genome. And that is the blueprint of every cell and organism or even species. That's the blueprint. So, what we are going to learn from now on is trying to see how these how this expression occurs. How does transcription occur? How does translation occur? And so on. So we will, and also in the later part, in the last unit, we will learn about when should expression occur and how much should expression occur. Right? It is regulation of gene expression. So as I was telling, this lesson is huge in a way, but we will learn quite a lot of things. But what I want you to pay attention to is the chemistry of polymerization, RNA polymerization, or transcription, and the type RNA polymerases, and also the mechanisms. Mechanisms should include these initiation, elongation, and termination. There will be fine details. I would want you to know the fine details because there is logic in every step of things. If you, as I was telling in the first class, I think you are learning the information system of life. That's molecular genetics. And in that case, in that sense, each step is highly logical. And if you want to appreciate biology or molecular biology, you need to look at the logic of why that is happening. You try to reason things. And that is what is this subject about. So your approach to this chapter also, like other chapters, should be looking for logic and try to remember a few specific examples. Okay. In today, we are going to discuss about overview of molecular genetics. That is what I was trying to do. We will kind of review, and then we will also discuss about overview of transcription. So you know the central dogma in molecular biology, of molecular biology, and you have three different components, major components, you can say. DNA, that is the master copy of what we have, the genetic information, from which through the process of transcription, we generate RNA, and through the process of translation, we generate protein. Okay? So there are certain other steps. Like, I mean, this is, in a way, one would be tempted to see it as unidirectional flow. This is something like unidirectional flow. But there are two more things. One thing, of course, is DNA replication. Of all these things, the DNA replication is the top, first priority. Okay, if DNA replication is not there, the cell will be as such. There will not be any more copies of it, and eventually it will fade away. That's what what will happen. So if you consider that if you count from the time of DNA as the genetic material, then DNA replication is the first evolved stable mechanism that is involved in replication. But again, we have a question about whether DNA is the first evolved genetic material or it is the RNA. We know we right now our process about we believe in RNA world in the sense that all of life or the genetic information in the beginning has started as RNA, and which is taken over to be the genetic information by DNA. That is because RNA supposedly has capacities to store information, as in the viral some of the viruses. It can act as enzymes, as in the case of ribozymes. We will have a look at it some other time. And then we have RNA as the messenger, which is not replaced. But then what you see is all the cellular life forms. Any cellular life form you see, the DNA is the genetic material. If you're counting from there on, you should think of it as DNA replication is the most important one, because without which there will not be any more copies of the cell. Okay? And then we have transcription. That's an one, important one, but it is only to produce RNA. What is the difference between RNA and protein? If we were talking about a language or a medium for conveying some information, we want diversity. And if you see that proteins have 20 different types of amino acids, so the combination of things is huge. In all of genetic information, you should also think of it as, what is information? It is about the sequence. Sequence of what? In nucleic acids, you count in terms of bases. If you are talking in terms of proteins, it is in amino acids. If you see the structures, the three-dimensional structure a nucleic acid can form, and compare it with the three-dimensional structures amino acids can form, is too huge. Amino acids have 20. There are 20 different types of amino acids. Some of them polar, some of them non-polar, some of them basic, some of them acidic, and so on. So their interactions will give rise to diverse types of structures. You can think of many different diverse structures, and also number of amino acids. You have very complicated and diverse proteins. And from that structural basis, you may develop a function. Function? What is a function? It could be either becoming a structural entity like microtubules or actin filaments, or it could be channels, like in the for membrane transport across the membrane, or it could be enzymes like hexokinase and so on. And that is why proteins have a unique proteins. The evolution of proteins has evolved, or you can think of it, has allowed the evolution of so many complex cellular forms. Okay. So we also learned about reverse transcription in where when we were reading about retrotransposons, for example, where using RNA as template, DNA is synthesized. You can also call it as RNA-dependent DNA polymerase. And there are, in many viruses, you will also find that they have, in retroviruses, the viruses that have RNA viruses, you have, if they have to replicate, then they use RNA replication. So that is also one of the points. Can we generate, is there a possibility for generation of protein-dependent RNA polymerization? That is something like reverse translation. So far, it hasn't been possible in the lab. Okay, it's not in the lab. It's not possible, and we haven't seen any examples in the nature that can make RNA from proteins. Probably it is useless, or it is too expensive, or it is not required. Okay. You will also find some examples where proteins can be synthesized without RNA, and that is called as non-ribosomal. Non-ribosomal, you can Google it, ribosomal protein synthesis. But they some of the antibiotics and others, some molecules are synthesized, or peptides, which are maybe 4, 5, or 6 peptides, and they can be synthesized without independent of ribosome. There are certain proteins, and those enzymes can synthesize smaller peptides. Okay, but that's possible. I hope you got an idea about what we are actually dealing with in this whole course. And I also want you to pay attention to this, where do these different events occur. If you take a bacterium and it has a genome, so it is assumed that it is the genome or chromosome of bacterium, which is called as the nucleoid. You would call it as nucleoid, right? Bacterial chromosome. And transcription will generate mRNA, and mRNA would be made into, this is cytoplasm, where from transcription you will get mRNA. From there you will get the protein. All of the events that happen in bacteria are happening in cytoplasm, because there is no nuclear membrane, or you don't find intracellular membrane systems within E. coli or within bacterium. So everything happens in cytoplasm. But if you take in eukaryotes, it is different. The eukaryotes have cytoplasm, and they have nucleoplasm or nucleus. This will be the nucleoplasm. And you also have a nucleolus. Right? There are 3 different structures that are directly related to what we are, to central dogma. So replication happens, you can think of all nucleic acid polymerization, right? All nucleic acid polymerization. When I say that, it includes DNA polymerization and RNA polymerization is happening in the nucleus. Okay, so replication and transcription is happening in the nucleus. In eukaryotes, as we learned in our 3rd chapter, or genomic or organization, we learned that majority of the eukaryotic genomes have, or genes have, a lot of introns. What are introns? Introns are called are intervening sequences. If that is the coding region, you have certain regions within the genome that are intervening or non-coding. So this would be called as the exon, and this is exon 2, and this is the intron. If expression of a particular protein should happen from the said gene, then the intron should be eliminated, and the elimination of the introns is called as splicing. Splicing of exons, that's how you should always remember. It's not removal of introns. When you say splicing of exons, you're saying joining of exons, which would mean that the introns are removed. Okay. So splicing also occurs in the nucleus, right? This RNA processing that is there, that also occurs within the nucleus. That mature mRNA that is formed is exported out to the cell, and their ribosomes will translate the mRNA. So you can think of all of protein polymerization or amino acid polymerization occurs in cytoplasm. There is one more thing that we need to think of, and I would like to use the example of ribosomes. Where did ribosomes come from? And what are they composed of? Ribosomes are composed of rRNA. There are 3 of them. And there are also many ribosomal proteins. Let's just to review what we are learning about, where things happen, where these events happen within the cell. we will take the example of ribosomes. And ribosomes consist of rRNA and ribosomal proteins. And where are they organized? They are organized in nucleolus, right? They are organized in nucleolus. When we read about translation, we will also learn, I think I have given it as an assignment. I have to look at it. There is a particular order of how the ribosomes are made, right? Which one is first put, RNA, and upon which protein has to add, and so on. And in my view or opinion, I think the ribosomes are the most complex things that I have ever seen in biology, because they can read nucleic acids and make sequence of amino acids. They can make proteins with a specific sequence. How can such a complex structure with a complex function arise? And you also will be interested to know that ribosome is able to kind of proofread itself to do to the best possible sequence it will add. So for me, ribosome is always a passion about trying. It creates wonder about how such things have come about. Okay, so let's see what happens to how ribosome actually has to can be made within the cell, biogenesis in a way. So think of some genes are there. This is the, say, rRNA gene, and there are more than 30 or 40 proteins, ribosomal proteins are also there. So assume that I'll put as R protein gene, ribosomal proteins gene. Okay, so I've just indicated one each for ease of understanding. First thing it has to do is something like this. It has to transcribe this gene, produce mRNA, and that mRNA should be spliced, and that mRNA should be carried out of the nucleus. In this cytoplasm, the already pre-existing ribosomes will synthesize the ribosomal proteins. Okay, these ribosomal proteins should again be taken into the nucleus. That is where rRNA genes are being transcribed. Okay, nucleolus is the primary structural thing where ribosomes are organized. That means all the RNA transcription happens in nucleolus. And the ribosomal proteins that are made should be carried back into the nucleus, and then ribosome subunits, right? In eukaryotes, you have 80S subunits. In prokaryotes, you have 70S subunits. In prokaryotes, it's a different thing, but I think to understand eukaryotic one is much more complicated, so it's better. So it has 30S and 40S and 60S subunits, eukaryotes. And those subunits, small and large subunits, will then be carried out to this cytoplasm, which will then be utilized to translate many other mRNAs. Okay? I hope you understood what I want to convey. And next, I also want you to understand a few words before we go. And this is terminology. Some of them I already covered. Genetic code. I'm seeing if it is there or not. Okay. Genetic code. What is a genetic code? If I was talking about translation, you have to, I'll just put ATG, GAC, CAG, CCC, GGG, and TGA. So the first one, this is always ATG. It is the start codon. TGA is a stop codon. When you say about genetic code, for the moment, you remember that you have to read the basis in basis of 3, and that is continuous. You cannot have any gaps, and it should always start with the genetic code or the open reading frame together. You should. There are. Let me finish the genetic code itself. Okay. So ATG is the start codon, and there are 3 stop codons. That is TGA, TAG, and TAA. So there are 3 stop codons and 1 start codon. So there are about a total of about 64 codons, out of which 1 is start, and there are 3 stop codons. The remaining 60 code for amino acids. Genetic code is degenerate. What does it mean? It means that one amino acid can be coded by more than one codon. Okay? One amino acid can be coded by more than one codon. That's what is meant by genetic code is degenerate. If I have to say one codon can code for more than one amino acid, is it true or false? It is false. One codon will code only for one amino acid. One amino acid can be coded by more than one codon. That's because we have about 20 amino acids and we have 60 codons. You can call it as 61 because the start codon also codes for something. So 61 codons code for, there are 61 codons, there are 20 amino acids. So obviously we have to have some of the amino acids coded many times. Okay, that's about some of the things about genetic code. And then we have, what is an open reading frame? You have a reading frame means it is about what ribosome does. Can ribosome read? When you say read, it means the product should be a protein synthesis. A protein should be made. Only then you say it as reading frame. So if a ribosome has to start protein synthesis, it requires ATG. That is the start codon. This is the first time an amino acid will be added. And then it will keep reading in steps of 3, and that is each one is a codon. So start codon is also coding for one amino acid, methionine. Then you have codon 2, 3, 4, and 5. It keeps on going until it encounters a stop codon. Any of these 3 stop codons can be there, and the presence of it, that is where the protein is 1, 2, 3, 4, 5, and that is where stop. Now you have a protein. That is an open reading frame. Okay. There are also, I also want to, I think I will not discuss about introns and exons now, because we already have discussed that. What I want you to also know now is about what is meant by plus strand and what is meant by minus strand. So I will try to write a sequence. Let's see how it goes. And so I'm writing an open reading frame. ATG, TAC, CAT, CCC, GGG, and TAA. Now I'm going to write the complementary strand of it. GGG. Right? If transcription occurred, if this one, the top strand acted as a template, what will be produced? If the bottom strand acted as a template, what will be produced? That's the question. Always remember, polymerization happens from 5' to 3'. Within the assignments, I have noticed that some people are saying it as 3 dash and 5 dash. In my opinion, I think that is wrong. This is a dash. Okay. If you said 5 dash, this is what it would mean. I think this is correct. 5'. Somewhere somebody must have used it as dash, and we are all carrying it that way. So I'm speaking according to the textbook. So 5'. This is probably correct one. So try to use the terminology right and say it as prime. I think I already told you this before. That's all right. So the bottom strand, if the bottom strand acts as a template, when we read about translation, we will also know those things in detail. If it acts as a template for translation, we would get a DNA like this: A, U, G, because it is RNA. So there is no thymine, you will have U instead of thymine. So UAC, CAU, CCC, GGG, and UAA. Right? That will be from 5' to 3'. This is the mRNA sequence that you're seeing now will be produced only if the lower strand acted as a template. Okay. If the top strand acted as a template, you would get something like this one. And that's not useful for us because it doesn't have an open reading frame. Right? There is no, we have to read from 5' to 3'. Do you see a ATG? Here is ATG. That could be a start codon if it were produced, okay? And GTA, CAT. There is no stop codon. So we will also do some exercises, and that is one of the reasons why I'm giving you taking this class is for you to see and make a comprehension of all the things that you have learned so far, including the lesson on mutations. So what I want to emphasize here is you should consider, because the mRNA that you're seeing now has the codons readable, right? It has the open reading frame, ORF. So that is called as the plus strand. Okay. The complementary of the mRNA will be called as the, or the one that doesn't have, that is the complementary to this ORF will be called as the minus strand. Okay. So if you are talking in terms at the level of RNA, then the bottom strand, is it similar to this or is it complementary to this? Compare the mRNA and the bottom strand. If you see these two, this is plus, then it is complementary, the lower strand. So it must be something like a minus strand. This is just terminology. You don't have to worry about it. If you look at the mRNA and the top strand, they are same except for T replaced with U. So that will be the plus strand. When we are talking now about ORF and gene expression and so on, then sometimes we will use the terminology as plus strand and minus strand. All you want you to remember, if you want to remember plus and minus, plus strand is the one that is the sequence of mRNA, that is plus strand. You can also remember it as the strand in which the ORF is readable or is present as it is called as the plus strand. If I gave you just these two strands and told you figure out the plus strand in that particular region, okay, then you will say the top strand is plus. Again, it is contextual thing. Say for example, I will try to write a sequence, and I don't know if I can make justice, but let's see. ATG, CAT, TAC, and TGA. I want to also write another one, ATG, right? There it is coming. Let me extend a little bit. GAT. TGA. I know what I'm trying to do. Let me check if I can make it. ACT. CAT and ATG. TAC. ATG. I'll change something here so that I won't confuse you. AAT, TCA, GTA. So what I'm trying to do here is I created a DNA molecule, right? A sequence like this. Try to look at where you can find ORFs in this. First thing, I'm giving you a very simplistic one, so it's easy to find out. One, ATG is the start codon, right? Then you have two more codons, and then you have a stop. Okay? No open reading frame will be this short. Usually proteins will be larger in size, at least some 20 and 30 amino acids and so on. But they can be, a larger protein can be processed to generate smaller peptides. That's a different issue. Now we are looking at 5 prime to 3 prime, and you have assumed that you have identified a start codon, ATG, second codon, and then third, and then I just put a stop codon there. Now look at the bottom strand, 5 prime to 3 prime. You saw an ATG, and you have next codons 1, 2. This is the start codon 1, 2, 3, GTA 4, and then you have TGA, that is fifth one, that is the stop codon. Now, if I'm talking about, I'll call this as ORF1, okay, and this one I'll call as ORF2. To produce a template for ORF1, I need the bottom strand to act as template. Only then you will get mRNA that is similar in sequence to this. If I want a sequence, if I want the expression of the lower strand, I mean the ORF to be expressed of the lower strand, then the top strand should act as template. Don't worry if you feel confused. We will look at these in the next class with more details. But right now, what I want you to understand is about trying to understand plus and minus. As I said, the use of the terminology plus and minus is contextual. If I'm talking in the terms of ORF1, then which one is plus? This will be the plus strand, and the bottom strand will be the minus strand. Just this will be the plus, and the bottom strand will be the minus. If I'm talking in terms of ORF2, if that is the context at that time, then this is the lower strand is plus and the top strand is minus. Just remember them like that. We will get to details in the next class. So I hope you understood a few things about it: genetic code, open reading frame, plus strand, and minus strand. I will try to use this one slide before we move on. I think this is what will give you the overview of what I was trying to describe also. So here is the DNA duplex. It is given as coil, so you might or you might not understand, but that's all right. In this, if you focus just in this location, you will see that this is the template strand. DNA, that is the template strand. That is template, it is minus. Okay? Because it is producing sequence that is present on this one. Right? The RNA is similar to the top strand. It's similar to top strand and it is complementary to bottom strand. Say from the point that I've already said, you can mRNA is coding because it has the open reading frame. mRNA is plus strand. It's not minus because the ORF is present. Because ORF is present and it's going to give rise to a complete protein, also we will call it as sense. Okay, this is a small exercise for you to, if you know how you describe mRNA, then you know everything else. mRNA is coding strand, coding. mRNA is plus, and it is sense. Anything complementary to it, you can call in that context as non-coding. It is called as minus, and it is called as antisense. So that is where the top strands and bottom strands, if you're trying to compare them at the level of DNA, then you would say this is the template. One important difference that you need to know is the bottom strand is template in transcription. That is how you generate the RNA here with the readable ORF. So the bottom strand here is the template. It is the non-coding strand because it does not have the ORF. It is actually having the complementary of the ORF. So you call it as non-coding. For the same reason you call it as minus strand, you can also call it as antisense strand. We don't call at the level of DNA, we don't call them often as antisense and so on, but it's good to know how to differentiate or how to recognize which one is plus strand, antisense strand, and so on. The top strand is the non-template strand because that is not acting as the template during transcription. But it is the coding strand because it has the ORF, and it is the plus strand, and it is also the sense strand. Okay, I'll stop here, and if you are following along with me, I'd like you to read the textbook and come and watch the animation that is already there with you. I think I have given access to the Watson animations. You please have a look at it and come back, and we can have a very good discussion. I'm trying to look at what has happened with the assignment. So the next assignment, if I remember correctly, I gave it for splicing or the transcription, right? I have given transcription assignment. So please try to do it now. If you are doing it along with me, then you will actually finish the assignment properly and well, right? So try to read the textbook. So today is 24th. In 3 days, I think you have to submit it. So tomorrow, yeah. I have given enough introduction for you to go through the textbook. And in the next class also, I will discuss some of the events, but I hope you will be able to do it along with me. You please understand that all the assignment exercise is to build your confidence, and I think some people have improved dramatically, and it's a very proud thing for me. And one more skill that I want you all to have is the ability to read the textbook, to read and enjoy it, right? We all like instant gratification. We want to do something, we want reward for it. Can you keep reading and for a reward that will come some long time later? Can you still keep working for it? That is delayed gratification. I think we should try to become accustomed to that, or be able to derive fun out of reading the textbook. Now we are doing this. The assignment is there, and we are reading the same chapter, and you have the book. either physical copy or a soft copy. And if you read along with me and along with the class, you will be able to do the assignment well, submit it on time, and you will get your 5 marks. If you think like that, it's always a win-win situation if you're doing it along with now. Try to read the textbook a little bit today, reread it tomorrow, and then you can come to the class. I hope you will understand the much better, and then you will record your assignment and submit it. That way your confidence will be tripled so that you can perform better and you will understand better. So it's all about you growing with skills. These skills are important for lifelong. You may forget about transcription. I don't mind about it. You may never use about transcription ever in life. I don't mind about it. You will gain the confidence and you will gain the ability to read and comprehend with delayed, even though there is delayed gratification. And that is the skill that will take you or will get you a long way in the future. So please read the text along with me for the next 3 days, and your assignment can be done much better and well. Okay? And I will not give the access to the video until the assignment deadline is over. So keep that in mind. Okay? Take care, and we'll see you in the next class. But any questions?